Every living organism is composed of chemicals which can be either elements or compounds. The elemental composition of living tissues can be determined by following a technique known as elemental analysis. Interestingly, we obtain similar elements while doing an elemental analysis of different living and non-living things. For example, we find that elements such as carbon, hydrogen, oxygen are common to the Earth's crust as well as living tissue. However, the composition of these elements is much higher in a living organism compared to the Earth's crust, as shown in this table. To ascertain the chemical composition of organic compounds in a living organism, we perform chemical analysis. For example, to find the chemical composition of a living tissue, such as a vegetable, we first grind it in trichloroacetic acid. As a result, we obtain a thick slurry. Now we strain this slurry through a cheesecloth to obtain two fractions. One fraction is the filtrate or the acid-soluble pool, and the other fraction is the retentate or acid-insoluble fraction. The acid-soluble pool contains organic compounds. Once a compound has been extracted, the next stage is to separate one compound from the others using separation techniques such as chromatography and centrifugation. Thereafter, various analytical techniques are used to identify the molecular formula and the structure of the compound. All the carbon compounds we derive from living tissues are called biomolecules. For example, carbohydrates, lipids, amino acids, water, and nucleic acid. Biomolecules such as amino acids and sugars are also known as metabolites because they are intermediates and products of metabolism. They are classified as primary and secondary metabolites. Compounds such as hydrogen, carbon, oxygen, nitrogen, sulfur, and sodium, which are involved in normal growth, development, and reproduction, are called primary metabolites. They are found in animal tissues. On the other hand, compounds such as alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, and spices are known as secondary metabolites. They are found in plant, fungal, and microbial cells and perform an important ecological function. Some of them play a helpful role as antibiotics and pigments. Living beings are also composed of inorganic compounds such as sodium chloride and calcium carbonate. To analyze the constituents of inorganic compounds, we carry out the following process. We take a small amount of living tissue, such as a leaf, and weigh it. This weight is known as wet weight. Then we dry this leaf to allow the water to evaporate to get its dry weight. Now, when we burn the leaf, the carbon compounds get oxidized and the leaf releases carbon dioxide and water vapor. The remaining ash contains inorganic elements such as calcium, magnesium, and potassium. Therefore, Analysis of compounds helps us identify organic and inorganic constituents in a living tissue. It also helps identify functional groups such as aldehydes, ketones, and aromatic compounds. In biology, however, functional groups are present as amino acids, fatty acids, and nucleotide bases, together known as biomolecules. Amino acids are the building blocks of all proteins. The amino group and carboxylic group are present on the same alpha carbon atom of the amino acid. Hence, they are also known as alpha amino acids. Amino acids are substituted methanes. There are 4 substituent groups occupying the 4 valency positions. These groups are hydrogen, the carboxyl group, amino group, and a variable group also known as the R group. Based on the nature of the R group, there are many amino acids. However, there are 20 standard amino acids in proteins. For example, glycine, in which the R group is hydrogen, alanine, where the R group is a methyl group, and serine, where the R group is a hydroxymethyl group. The chemical and physical properties of amino acids are essentially of the amino, carboxyl, and the R functional groups. As amino acids contain both the amino group and carboxyl group, they can be acidic, such as glutamic acid, basic, as in the case of lysine, and neutral, like valine. Similarly, there are aromatic amino acids such as tyrosine, phenylalanine, and tryptophan. A unique property of amino acids is the ionizable nature of the NH2 and COOH groups. Amino acids behave as both bases and acids. In solutions at a particular pH, ions with both positive and negative charges are equal. This form is known as the zwitterionic form. Another group of biomolecules is fatty acids. Long chains of fatty acids and their derivatives lead to the formation of lipids. Lipids include a broad range of molecules such as fats, waxes, fat-soluble vitamins, and glycerides. They are generally insoluble in water. Fatty acids that form lipids have a carboxyl group attached to an R group. The R group could be a methyl, ethyl, or a higher number of CH2 group with 1 carbon to 19 carbons. For example, palmitic acid has 16 carbons, including a carboxyl carbon. Fatty acids can be saturated or without a double bond. They can be unsaturated or with one or more C double bond C. Glycerol or trihydroxypropane is a simple lipid. When fatty acids combine with glycerol, they form esters, also known as glycerides. Glycerides can be monoglycerides, diglycerides, and triglycerides. Based on their melting points, glycerides may be either fats or oils. Oils have a lower melting point and hence remain as oil in winter, while fats freeze easily at low temperatures. Lipids, that contain phosphorus and a phosphorylated organic compound are known as phospholipids. They are found in the cell membrane. Living organisms also have many carbon compounds with heterocyclic rings, such as adenine, guanine, and thymine. These compounds are called nucleotide bases, and they are important biomolecules. When carbon compounds with heterocyclic rings are attached to a sugar, they are called nucleosides. For example, adenosine and thymidine. On the other hand, if a phosphate group is also found esterified to a sugar, they are called nucleotides such as adenylic acid and thymidyllic acid. Nucleic acids such as DNA and RNA consist of nucleotides. They are molecules involved in heredity. Thus, biomolecules act as building blocks of life and perform vital functions in living organisms. All living organisms produce organic molecules called biomolecules. Biomolecules can be broadly divided into two types: biomicromolecules and biomacromolecules. On performing a chemical analysis, we find that biomicromolecules are chemical compounds found in the acid-soluble pool with molecular weights ranging from 18 to 800 daltons. Some examples of biomicromolecules are water, minerals, amino acids, sugars, lipids, and nucleotides. On the other hand, biomacromolecules are compounds found in the acid-insoluble fraction. They have molecular weights in the range of 10,000 daltons and above. Biomacromolecules, or compounds which come from the insoluble fraction, are proteins, polysaccharides, and nucleic acids. Interestingly, lipids, which are biomicromolecules, are also found in this fraction. The acid-soluble fraction represents the cytoplasmic composition, while the acid-insoluble fraction consists of macromolecules derived from cytoplasm and organelles. Together, these two fractions represent the complete chemical composition of living organisms. Biomacromolecules have molecular weights in the range of 10,000 daltons and above. Let's understand why lipids, whose molecular weight does not exceed 800 daltons, are a part of the acid-insoluble fraction. Lipids have a small molecular weight, and they are arranged in the cell membrane and in the membranes of mitochondria, chloroplast, and Golgi apparatus. During a chemical analysis, when we grind a tissue, the cell structure ruptures and various membranes break into pieces and form vesicles. These vesicles are not water-soluble because lipids are hydrophobic. So, they get separated along with the acid-insoluble pool. We'll now learn about polysaccharides in the acid-insoluble fraction. Polysaccharides are long chains of sugars. They are thread-like structures consisting of different monosaccharides as building blocks. In a polysaccharide chain, for example in starch, the right end is known as the reducing end, while the left end is called the non-reducing end. Polysaccharides can be divided as homopolysaccharides or heteropolysaccharides. When all the monosaccharide units in a polysaccharide are of the same type, it is called a homopolysaccharide or simply homopolymer. Some examples are cellulose, starch, glycogen, and inulin. Cellulose is a homopolymer consisting of only glucose as a monosaccharide unit. Some examples of cellulose are a plant's cell wall, paper made from plant pulp, and cotton fibers. Starch, on the other hand, is a homopolymer of glucose which acts as the storehouse of energy in plant tissues. Starch forms helical secondary structures, and in that position, it can hold iodine molecules. Conversely, cellulose cannot hold iodine molecules as it does not contain complex helices. In animals, glycogen acts as a homopolymer formed of glucose as a monosaccharide unit, a variant of starch. Inulin is a polymer of fructose. Polysaccharides, which are formed of more than one type of monosaccharide unit, are called heteropolysaccharides or simply heteropolymers. They are complex in nature, with amino sugars and chemically modified sugars as building blocks. Glucosamine and N-acetyl galactosamine are examples of heteropolymers. Another type of biomacromolecule in the acid-insoluble fraction of living tissues are nucleic acids. A nucleotide acts as a building block of nucleic acid and forms a polynucleotide chain. A nucleotide has three chemically distinct components, namely a heterocyclic compound, a monosaccharide, and phosphoric acid or phosphate. The skeletal heterocyclic ring compounds are actually nitrogenous bases such as adenine, guanine, uracil, cytosine, and thymine. There are two types of nitrogen-containing bases, namely purines and pyrimidines. In a purine, 6-member and 5-member nitrogen-containing rings are fused together. Pyrimidines, however, have only a 6-member nitrogen-containing ring. Therefore, adenine and guanine are substituted purines, while uracil, cytosine, and thymine are pyrimidines. The sugar found in nucleotides is either ribose or 2'-deoxyribose. Both these are monosaccharide pentose sugars. Therefore, nucleic acid containing deoxyribose is called deoxyribonucleic acid or DNA, while the one with ribose is called ribonucleic acid or RNA. Both DNA and RNA carry genetic information and are found in the cells of all living things. Together, polysaccharides, nucleic acids, and polypeptides or proteins form the macromolecular fraction of all living tissues. Proteins are important biomolecules needed by all living organisms. They are polymers of amino acids arranged in a linear chain and linked by peptide bonds. Therefore, they are also known as polypeptides. Amino acids that go into the making of proteins can be essential or non-essential. essential amino acids are found in our diet, while non-essential proteins are synthesized by our body. There are 20 types of amino acids that come together to form proteins, and so they are also known as heteropolymers. In a heteropolymer, there are different types of monomers repeating n number of times. Proteins form very interesting three-dimensional structures. The structure of molecules is depicted differently in different disciplines. In chemistry, structure means the molecular formula. For example, salt is also known as sodium chloride or NaCl, where NaCl is the molecular formula and not the structural formula. On the other hand, organic chemists present a two-dimensional view of molecular structure. For example, in benzene or naphthalene, this structure represents the structural formula, whereas the molecular formula is C6H6. Physics considers a three-dimensional view of molecular structure. However, biochemists have described the protein structure at 4 levels: primary, secondary, tertiary, and quaternary structure. Let's learn more about them. The primary structure is the simplest structure, which refers to the positional information of amino acids in a protein. Hypothetically, a protein is a thread formed by amino acids where the left end is represented by the first amino acid or the N-terminal amino acid, and the right end is represented by the last amino acid or the C-terminal amino acid. The protein thread is represented as a horizontal straight line so that the position of the amino acids can be determined. However, in nature, proteins are not completely straight but are folded in the form of a helix, somewhat like a revolving staircase. In the secondary structure, amino acid links get longer, and due to the heaviness of the molecule, it tends to bend. This preliminary bending is considered to be the secondary structure. It is either an alpha helix or beta sheets. The third level of protein structure is the tertiary structure. It refers to the three-dimensional structure of the entire polypeptide chain. The function of a protein, except as food, depends on its tertiary structure. If this is disrupted, the protein is said to be denatured and it loses its activity. The fourth level of protein structure is the quaternary structure. This structure is formed by several protein molecules or polypeptides, also known as protein subunits. These subunits are arranged as a linear string of spheres arranged one upon each other in the form of a cube or plate. For example, adult hemoglobin consists of 4 subunits, 2 of which are identical to each other. Therefore, 2 alpha-type subunits and 2 beta-type subunits collectively form human hemoglobin. Some of the most common proteins we come across are collagen, rubisco, trypsin, insulin, antibodies, receptors, and GLUT4. Let's learn more about each of these proteins. Collagen is the most commonly occurring protein in the animal world. Collagen fiber is generally found in connective tissues and acts as an intercellular ground substance in the space between the cells. On the other hand, ribulose bisphosphate carboxylase oxygenase, or Rubisco, is the most abundant protein in the whole of the biosphere. It acts as a catalyst during the process of carbon fixation. Trypsin acts as an enzyme, and insulin acts as a hormone. Another protein called antibody helps fight infections. Receptor, another protein, helps in sensory reception, while glucose transporter 4 or GLUT4 helps in transporting glucose to the cells. In this manner, various proteins play a vital role for every living organism. The structure of these proteins is crucial in determining their functions. Bonding plays a crucial role in the making of a polymer. A polymer is formed when many monomers come together and bond in long chains. For example, polyethylene is made up of repetitive units of ethylene monomer. Let's look at the bonding in biomolecules such as proteins, polysaccharides, and nucleic acids. Proteins are amino acids linked by peptide bonds. This chemical bond is formed when the carboxyl group of an amino acid reacts with the amino group of the next amino acid, losing a water molecule. This process is also known as dehydration. Polysaccharides are formed when several monosaccharides link together by a glycosidic bond. This bond is also formed due to dehydration. The glycosidic bond is a functional group that can exist between two carbohydrate molecules or between a carbohydrate and a non-carbohydrate molecule, as between glucose and serine. The bonding in nucleic acids is different. Here, the 3' carbon atoms of one sugar of a nucleotide are linked to the 5' carbon atoms of the sugar of the succeeding nucleotide by a phosphate moiety. The phosphate and hydroxyl groups of sugar join to form an ester bond. The ester bonds formed by the phosphate group between the 3rd and 5th carbon atoms of adjacent pentose sugar rings are called phosphodiester bonds. This bond links 3' carbon atoms of one sugar molecule with the 5' carbon atoms of an adjacent sugar molecule. The phosphodiester bond is very vital to all forms of life, as it forms the backbone of deoxyribonucleic acid or DNA strands. According to the famous Watson-Crick model, the B form of DNA is the native conformation of DNA in solution. The B form of DNA is a metabolically stable configuration. Some of the features of bDNA are that it exists as a double helix. Two strands of polynucleotides run in opposite directions. In other words, they are antiparallel. That is, one strand runs in 3' to 5' and the other strand runs in 5' to 3' directions. The backbone of DNA is formed by a sugar-phosphate sugar chain. The nitrogen bases face inside and are almost perpendicular to the backbone. The 4 bases found in DNA are adenine, cytosine, guanine, and thymine, which are abbreviated as A, C, G, and T respectively. Each type of base on one strand forms a bond with just one type of base on the other strand. For example, adenine on one strand base pairs with thymine on the other strand by a double bond. Similarly, cytosine on one strand base pairs with guanine on the other strand by a triple bond. Each strand appears like a helical staircase, and every step of ascent is represented by a pair of bases. One full 360-degree turn of the helical strand would involve 10 steps or 10 base pairs. In other words, there are 10 phosphate sugar groups in every full revolution of the DNA strand. The distance along the helix axis corresponding to one turn is known as pitch. In a B DNA, the pitch is 34 angstroms. The rise per base pair, or the distance between two base pairs, is 3.4 angstroms. The different types of bonds we have studied are crucial as they provide structure and stability to biomolecules and make them functional.